Effects of potentially toxic elements on soil enzyme activities. The biological activity of soils is an essential parameter of their ecological status. The potentially toxic elements present in the soil due to anthropogenic activities disturb the normal functioning of soil biota and, hence, the entire soil system. As the concentration of potentially toxic elements increases, the activity of most enzymes is significantly reduced and may be caused directly by the interaction between the enzyme and the potentially toxic elements, which is not associated with a reduction in microbes. Potentially toxic elements influence the enzymatic activity, by destroying the spatial structure of the active groups of the enzyme, by inhibiting the growth and reproduction of microorganisms, which reduces the synthesis and metabolism of the microbial enzymes. Soil microbes and soil enzymatic activities are significantly correlated. Some enzymes secreted by microorganisms participate in soil ecosystems and energy together. Potentially toxic elements in enzymes play a triple function: catalytic, structural and regulatory. Zinc is an integral part of the number of enzymes and number of intracellular enzymes, viz., carbon anhydrase, carboxypeptidase, thermolysin, alkaline phosphatase, dehydrogenases, glyceraldehyde 3-phosphate, alcohol, glutamine, fructodiphosphate aldylases, superoxide dismutase, DNA and RNA polymerase, trinotransferase need zinc for proper functioning. When potentially toxic elements are present in excess, the natural functions of metals are distorted. Tejada et al., and Cheng, reported that the enzyme reactions are inhibited by potentially toxic elements in 3 different ways: 1. Complexation of the substrate. 2. Combination with protein active groups on the enzyme, and 3. Reaction with the enzyme substrate complex, while Vig et al. Reported that as potentially toxic elements interact with the enzyme active sites and substrate complexes, denatures the enzyme protein and competes with metal ions those are needed to form enzyme substrate complexes, inhibit soil enzyme activities. Nuaimi and Maktoum reported that potentially toxic element pollutants found in the soil can cause their deleterious effects by any of 4 ways: 1. There is oxidative stress in organisms due to the release of the oxy radical, which is produced by redox cycling of potentially toxic elements. 2. Proteins are deactivated or denatured as metals bind to sulfhydryl groups of proteins. 3. Antioxidant ability of cells retards as potentially toxic elements bind in intracellular glutathione and / or antioxidant enzymes. 4. Metalloenzymes became inactivated as potentially toxic elements compete for metal cofactor binding of metalloenzymes. Nuaimi and Maktoum also reported that the potentially toxic elements such as Hg2+, Chu2+ inhibited alkaline phosphatase enzyme more strongly than Cd2+, and CO2+, and also that alkaline phosphatase is readily inactivated with the exposure to potentially toxic elements. Uncharged at neutral pH, arsenic can diffuse across the cell membrane. It can inhibit the enzymatic activities even at low concentrations. Bhattacharya et al. Reported that arsenic reacts with a sulfhydryl group of the enzyme to form arsenic sulfide, resulting in the decrease of enzymatic activities. The decrease in enzymatic activity by arsenic may be due to 1. By interacting with the enzyme substrate complex. 2. By denaturing the enzyme protein, or 3. Interacting with the active protein groups. The influence of potentially toxic elements on soil enzyme activity depends on pH, nutrient form and amount, potentially toxic element concentration and availability, enzyme type, etc. Afoigbu et al. During their research found that there was the significant negative correlation with the potentially toxic element contents and the activities of dehydrogenase, polyphenol oxidase, hydrogen peroxidase, alkaline and acid phosphatases and urease. There was a negative but non-significant correlation between ZN content and dehydrogenase activity, CD content and hydrogen peroxidase and urease activities. Diana et al. Reported that at the brownfield LSB, extracellular soil enzyme activities are notably high at a site with the highest potentially toxic element loads. Soil had been unmanaged for over 40 years, left alone to naturally succeed, and there is a strong relationship between these enzyme activities and CR and V in particular. This study demonstrates the capacity of some potentially toxic element contaminated soils to enzymatically function well under seemingly restrictive conditions. Wytrovska et al. Found that dehydrogenases, Acid and alkaline phosphatases exhibited the highest sensitivity towards Zn, and it decreased in the order of metal concentrations, Zn greater than Cd greater than Chu greater than Pb. In contrast, urease was more tolerant to Zn. The sensitivity of urease was as follows: Chu greater than Zn greater than Cd greater than Pb. In respect of their sensitivity to concentrations of the bioavailable pool of Cd, Chu, Pb, and Zn, the enzymes can be arranged as follows: alkaline phosphatase greater than acid phosphatase greater than dehydrogenases greater than urease. Bonsall et al. And Bonsall during their studies found that potentially toxic elements accumulated in soils due to sewage water irrigation increase the activity of the enzymes dehydrogenase, acid and alkaline phosphatase, urease and catalase up to 14 days of incubation and decrease thereafter. Dirdzian et al. During their studies found that potentially toxic element pollution affects the activities of beta-glucosidase, chitinase, leucine aminopeptidase acid, phosphomonoesterase and acetate esterase enzymes in the soils. Gromykova et al. During their work reported that the increase of mobile potentially toxic element forms content in soil inhibited the cellulose-degrading and urease activities after 30 days of the input of metal into the soil. The inhibition of biological activity of the studied metals formed the following series: Cd greater than Pb greater than Zn greater than Chu. Yu and Cheng during their studies found that addition of Chu, Cd, and Pb firstly enhanced urease activity and thereafter it declines, while with the increased concentration of Zn the activity of urease declines. In addition to Chu, the catalase enzymatic activity initially enhanced and thereafter decreases. The catalase activity continuously decreases with the addition of Cd, Pb, and Zn. Addition of Chu, Cd, Pb, and Zn in soil results in a decrease in microbial biological carbon content. Kahn et al. Found that in the Cd and Pb treated soils the activity of acid phosphatase and urease was minimum after 2 weeks of incubation. The enzymatic activities of the acid phosphatase and beta-glycosides were lowest in the soil samples having the maximum concentration of potentially toxic elements. They also reported activity of acid phosphatase and beta-glycosides was significantly influenced by pH and non-significantly with soil organic matter. A review of the literature shows that sensitivity of dehydrogenases is Hg, 2 mg, greater than Chu, 35 mg, greater than Cr6+, 71 mg, greater than Cr3+, 75 mg, greater than Cd2+, 90 mg, greater than Ni2+, 100 mg, greater than Zn2+, 115 mg, greater than S3+, 168 mg, greater than CO2+, 582 mg, greater than Pb2+, 652 mg Kg-1, Chu2 greater than Zn2 greater than Cr6 greater than Hg2 greater than Ni2 greater than Cd2 greater than Cr3 acid phosphatase, Chu2 greater than Al3 greater than Cd2 greater than Zn2 greater than Fe3 greater than Ni2 greater than Pb2 greater than Sn2 greater than Fe2 greater than CO2 and alkaline phosphatase, Cd2 greater than Al3 greater than Zn2 greater than Fe3 greater than Chu2 greater than Pb2 greater than Ni2 greater than Fe2 greater than Se2 greater than CO2